time so that you just get there by reiteration, more or less. I'm going to talk to you about what happens inside a cell and the dangerous environment that the cell, center of the cell is. We'll talk a bit about what ATM does and then try and talk a fair bit more on neurodegeneration. I don't have time to go through all of this, and I really didn't want to talk for too long. So we'll continue with this discussion next time. And unfortunately, I'm not going to be able to get to telling you about the research, which is something I wanted to do, but I promise try and do that next time. So what I told you was, if you remember, we started talking about how you start off as a single cell, and that cell can become a whole human being. And so it's got to have all the information that's needed to make a human being. And that information is stored in a linear, long DNA molecule called DNA. The DNA has 4 building blocks, A, T, C, G, and these 4 building blocks are organized like beads on a string to give a strand, and then, if you like, the little beads. And they interact between themselves. So you land up with a molecule that's got 2 strands, double-stranded. And then there's these weak interactions between these building blocks. And A always goes with T and C always goes with G. And then that long ladder type structure is often twisted, looking like a spiral staircase. And this is the classical picture that you often see of your double helix, your DNA, which is a double helix. Then at the other end, we talked about the worker molecules, which are the molecules doing the task in the cell. They have 20 building blocks, and when they're made also in a linear array, but because they can interact a lot between each other, they form a 3-dimensional structure. And it's that 3-dimensional structure and the shape that makes any particular protein be able to do its particular task. But there's a protein factory that is making these proteins, but it can't use DNA. In fact, it uses a subcode that is made from the DNA called messenger RNA. This also has 4 building blocks. They're very similar to DNA, but subtly different. And one of the differences is the U instead of a T, and it's also single-stranded. And then, just a minute, how do I? I've tried that before, and it did. I seem to quite often have difficulty when I want to go to the next slide. Oh, there we go. Okay. So what I told you was that what you first do is copy your DNA to something called pre-messenger RNA. And that's an exact copy, but then from that you cut and space, because the information that you want here is not contiguous in your DNA. So you cut and splice, throw away the intervening bits and make the bits that are going to code finally for your protein. And that process is called splicing. I've forgotten what I did now to get to the next slide. Okay, so then I also told you that, so this is the kind of organization. Your DNA is used to make your messenger RNA subcode, and that's used to make your protein. I told you that this process here is called transcribing, and I want you to remember that. I put it in red because we're going to come back to talk about transcription. Basically, you can think of you're transcribing one code into another code, and then the protein factory makes your protein. Proteins can be remade, they're dispensable. So, but you can't make another protein from one protein, you have to go back here. Similarly with messenger RNA. So these both are dispensable, but what really matters is your DNA. That's what's critical, because that's the molecule that carries all the information to make that cell what it is. And a lot of resources are put in to maintaining the integrity of the DNA. And we call that maintaining genomic stability. And that is the role that ATM does. So ATM has a task in helping to maintain this DNA, the stability of your DNA. Okay, so that's the summary of where we were last week. Trying to bring out the points that we're going to be used to talk about today. Now, the inside of a cell is actually a dangerous place. And your DNA can get very readily damaged. As I told you, all your components, DNA, RNA, and proteins, all get damaged. But what really counts is your DNA. So the cell puts quite a lot of energy into trying to maintain the integrity of the DNA. One of the damaging agents are called reactive oxygen species. You may have heard them because they're quite often talked about. Sometimes they're called oxidants. And we can get something like 60,000 lesions per day to our DNA. These can be damages to those little building blocks, or and sometimes they can cause a distortion in this sort of strand, or they can be what we call single strand breaks, so damages in just one of these strands, and occasionally damages in both strands at the same point. And if this happens, and if there's sort of maybe something else going on here, you can even lose some of that DNA material and some of those building blocks. And that's clearly a problem, because that's what I told you and meant to repeat but didn't, is it's that sequence of those building blocks in the DNA that is essential for effectively who we are, and we all have a different sequence of our building blocks. And the most biologically significant lesion is a DNA double-strand break, and that's the lesion that ATM is acting or triggered by. And I just wanted to kind of reiterate that. So what I mean by a double-strand break, it's on both of these strands of your DNA molecule. Here it's the linear, or where it's as a helix. It doesn't have to always be at exactly the same place. So here they're sort of a bit staggered. And because these bonds are not that strong, that this tiny little one here may not be enough to hold the whole molecule in contact. So even quite closely spaced lesions can still cause that molecule to come apart. So we can think of a single strand break in one of the strands, or a double strand break, and I'm going to be calling them SSBs and DSBs. So reactive oxygen species are byproducts from our own metabolism. We generate them ourselves. They can be counteracted by antioxidants, and maybe you've heard of antioxidants in your diet. Often we're told we want to have antioxidants in our diet, and they mainly produce damage to the building blocks and single strand breaks, but not very often double strand breaks. And the reason is that they don't have that much energy, and relative to the kind of size or width of the DNA, they can't do damage to both strands at the same time. They can you can get double strand breaks arising from two overlapping, closely spaced single strand breaks, but by and large, you don't get those double strand breaks. But also, processes involving DNA are complex processes, and they can cause damage as well. And we're going to come back to talk a little bit more about that later. Damage can also arise from external agents, such as chemicals, ionizing radiation, and sunlight. And in the context of AT, ionizing radiation is particularly important, because ion radiation is like a track going through a cell, has a lot of energy, and so it can damage both of those strands. And that's why AT cells are very radiation sensitive. We don't want AT individuals to be giving radiotherapy, which is a big dose of radiation. X-rays and CT scans are much lower doses, and they're very important tools. So you do want to use those, but we recommend, or it's recommended, that they're used only when essential. Don't overdo it unnecessarily. Sunlight, UV radiation, produces a very different form of damage. It does damage your DNA. So AT individuals don't have any problem from sunlight. You don't have to worry about going out into the sunlight. There are individuals who can't repair this type of damage, and they do have to remain inside, and they get many skin cancers, but that's not an issue for AT. And so for the AOA1 family, I'll just say that I had a look, and as far as I can see, AOA1 deficient cells are not very sensitive to either ionizing radiation or to sunlight. Exactly whether that translates to what happens clinically and in the individual is a little bit more of a gap, but usually it's pretty good correlation. When we can see it in the cellular studies, then you would be sensitive, and the fact that we can't see it would suggest that wouldn't be the case. The other point is that occasionally cells very deliberately create damage in your DNA for certain processes, and that occurs during the immune response. I'm not going to go into that in any more detail today, but we'll try and touch on that another day. But that probably underlies this immune deficiency of AT individuals. So, yeah. Okay, so what I want to just do is go through now some of the processes, processes are more or less discussed with you, which involve DNA, and think how they might be impacted by the lack of ATM. So an adult is estimated to have something like 30 to 40 trillion cells. And if you think that's arised from that one single cell, that's an awful lot of times that cell has, or your cells in general, have got to divide. So when a cell divides, it's very simple. It's just like this. We have one cell, this was a single cell here, going through a phase and then dividing into two cells. And we usually call this the mother cell and the daughter cells. But when you think about it, I've told you we've got 23 pairs of chromosomes, and those pairs are not identical to each other. As AT carriers, you will have one chromosome 11 from one parent and the other one from the other parent, so they're not identical. And all your cells in your body have got an identical DNA content. So those 23 pairs of chromosomes have got to be equally divided into the two daughters every time the cell divides. And when you think about it, that's quite a complex process. And then prior to dividing, your DNA needs to be copied, because you need to be, for each daughter cell to have that same amount of your DNA. And that complicated process too. So that act of cell division going through the thing can be impacted by a double strand break. If it means you may lose some of that genetic material when you divide. But now we come on to this concept of trying to replicate your DNA. So this is Watson and Crick, who identified the structure of DNA, got Nobel Prize for it. And these are the ladders that are here. And it's not very easy to see, but there is a sort of strand going up here. And in many ways, they didn't actually do all the experiments to unravel this structure. There was Rosalind Franklin, who wasn't part of the Nobel Prize, who did a lot of this work. But what they were outstanding in realizing was that the structure of the DNA allows you to replicate, for it to be readily replicated. And basically, you've got this double helix, and because these bonds are quite weak, they can come apart almost like a zipper apart. And so effectively, you've got your double helix here, and I've said these bonds are quite weak. so they can come apart as shown there. And what that allows is that then you've got, and we call this the existing strand, the previous strand. You can then make a copying that exact sequence that you had before by just adding one more building block onto the other down here, and that will be an exact copy of what you had before. So this is a means of replicating your DNA with exactly that same sequence of building blocks that you had before. But errors can arise during this process, and sometimes mutations happen, and that's quite possible that in some ancestor, the way you got that original mutation in the ATM gene was when just a wrong building block was put in here during replication. But this was the special replication to make the germ, to make the sperm or the mother's egg. But also a double-strand break will prevent efficient replication. So here you can imagine if you've got break in both strands, that will harm how you're going to be able to do that replication. The other important transaction, this looks complicated, but don't panic. It's the point I want to get over is very easy. The other transaction that we've talked about involving DNA is that transcription. Remember, you're making your RNA from your DNA, and that RNA is going to be used to make the worker molecules, the proteins. And I told you the first step is to make an exact copy. You're only doing it for that one bit of information that is required for that one protein. To make an exact copy first, and then it's cut and spliced. And what really the point of what I wanted to show you here is that that requires your DNA being opened up. It's actually a very complex process, and it requires all the what we call the transcription machinery to get in here and copy these sequences. So DNA damage can prevent efficient transcription, but also DNA damage can arise during transcription. When I say damage, I often mean you've got very complex chemical reactions occurring here. Sometimes they go wrong. Sometimes they get stalled. So you can have little bits of machinery here that are kind of stuck or abortive or not quite right. So you can get things going wrong, or you can get damage in structures like this. The one other point I want to bring out here, and again, a horrible looking slide, but it's actually, the message is very simple. I've told you you've got 2 meters of DNA that's all got to fit in this one tiny little cell. And the cell has a way of really compacting that. And basically, that's what's shown here. This is your kind of very open DNA, like you've got it here. But it gets wrapped around little building blocks. They're called histones, but they're proteins. It gets wrapped around these and then assembled into a structure. I can't see it because you're all down here, but hopefully you can see it. All wrapped into this little structure and finally into making a chromosome. So your DNA is really, really compacted, and then it has to be opened up to replicate, to copy one strand from another, and it has to be opened up for this act of transcription. And interestingly, that compaction, or importantly, that compaction helps to protect the DNA from some of these DNA damaging agents in the cell, particularly reactive oxygen species. So when you're open up like this, you've got the added possibility for damage happening in this vicinity. Okay, so that's really, I've told you now, the dangerous environment inside a cell, but also the difficulties of some of these processes that are using DNA and how our damage can arise, how damage to our DNA can arise. So now I'm going to tell you what ATM actually does. And basically it changes proteins by adding a tiny little group to them. And this is called, it phosphorylates them. This little change is called the phosphate group, which is shown there, and the process is called phosphorylation. And the enzymes, proteins that do that are called kinases. You may have heard us talk about the ATM kinase or the ATM kinase activity. It's that activity that just adds on this little tag to this protein. Don't worry too much about here. This could be any kinase, and this ATP to ADP just means it uses energy, but that's not relevant for what we're talking about. Now, what can this little guy do? Well, you remember me telling you that proteins fold, and that it's those interactions between the amino acids that make how they fold. And so this slide was a bit exaggerated, but it shows you how this little P, this little phosphate group, can change how this folds, mainly because it can affect size or charge, or something like that. And that little change in structure can alter the way the whole protein functions. It may help it bind something, may stop it binding something. It may even make it get destroyed, because there are proteins in the cell that recognize certain shapes and destroys them. So I kind of tried to think of a load of different analogies for this, and I came finally to think of it as being in a lockdown specific situation. And in a way, what I was thinking of is that you might want to lock down, particularly the hospitality industry, and change that so that the hospitality industry can't function, but everything else can function. And you don't want to, you want to pick on those individuals, if you like, which are doing specific tasks that may make it more dangerous for if we're all interacting. So important point is that ATM is activated by a double-strand break. So it only makes these changes when a double-strand break is there. And for that reason, it's called a damage response kinase. And it can modify over 1,000 proteins. Not any one cell won't have those many modified, but it's capable of doing that. And there's just a certain little marker, much like the person being locked down may, if in the hospitality industry, may have an apron. There's a few little amino acids that kind of enable it to be tagged. So it can recognize certain proteins and will then bung this tag on them. There are many kinases in the cell, quite a number of kinases in the cell, but there's in fact only 2 that are activated by a double-strand break. And they have what they recognize is very different, but the other one that is recognized is in the process that is repairing these double-strand breaks. But there is another type kinase called ATR, which stands for Ataxia-Telangiectasia related, very related protein that can tag many of the same proteins that ATM can tag. So there is some redundancy, but importantly, it's activated by a very different lesion. It's not activated by a double-strand break. There is some crosstalk. Sometimes, I just don't want to go into what lesion activates ATR, but sometimes the double-strand break can get converted to a lesion that activates ATR and vice versa. And I think somebody last week asked me about redundancy and if there's any other molecules that can do the same thing as ATM. And so that's why I kind of brought that one in there. So now, what is ATM doing at a double-strand break? And I think it's best to think of it as a policeman than as the doctor doing the repair. So it coordinates a whole response to a double-strand break by sending signals, by tagging those different proteins and slightly changing their function, or dramatically changing their function in some cases. So this was the best, unfortunately it's an American cop, but it was the best picture I could get to kind of display that. it's not doing the repair itself, but this is where I wanted specifically to say about AOA1, that is a repair enzyme. It's a specialized sort, and it actually will function at both a single strand break and a double strand break. But it's doing the repair itself. It's not doing all this signaling. So some of the things that ATM is doing in the cell, those signals it's selling out will not be done by, so the protein defective in AOE1 is called aprataxin, and it won't be done by aprataxin. So what this does, what these signals can do, can stop some of the transactions on the DNA, and particularly sometimes near the double strand break. So it's like there's a major accident and it's saying, stop, don't crash into that accident and make the whole thing worse. It will stop replication. It stops cells dividing. And I've explained to you how those are things that are bad news if you've got a break in the cell. It stops transcription, but not everywhere. So it's making RNA to make the proteins. And sometimes the proteins it's making might be required to actually help with the repair. So you don't want to stop everything, but it very cleverly stops transcription right near where there's a break. And that's to stop those two machineries kind of coming together. So it stops the accident getting any worse, and it promotes good recovery. Now, the other thing that it can do, which is really interesting, is it activates a pathway of cell death, and it's often called a suicide pathway. And I put a gravestone here and sort of said it sends body to the mortuary. And the rationale there is that we're an organism. We're a whole organism. We're not just a collection of single cells. And the body doesn't want to have non-functional cells in it, bad, broken, damaged cells. So it would be better off getting rid of them, and it activates this cell death pathway. The other important thing is for cancer, because a badly damaged cell has the potential to be misrepaired rather than accurate repair. And I'll come back and talk about that a little bit later. But by getting rid of cells that are very badly damaged, you diminish the risk of getting cancer. And obviously that's important for thinking about what ATM does. It also helps to compact the, decompact and compact the DNA around where there's a break. So you can think of this as like an accident in a forest. The doctor can't get there because there's too many trees around. So it helps collapse the trees, open up, so the doctor can get to the accident, but it's done in such a way that it can put them back again. And in fact, it very cleverly decompacts a little bit, it kind of leaves the middle open so that doctor can get there, and it'll decompact all around it to stop erroneous repair taking place. And just one final thing that ATM does is it senses, and this is a slightly different way it does this, but it senses what we call oxidative stress, which is it's sensing those reactive oxygen species. And then it'll regulate antioxidants. So if there's a lot of damage, a lot of reactive oxygen species, it will help produce more antioxidants to counteract them. And the really critical thing of all of these is it helps those double strand breakthrough being repaired. And that's important because if they're not repaired, they're lethal. But very important, but it's not the repair, it's not the major doctor. The repair tends to take place. There's not much of a repair defect if you don't have ATM. But the really important thing is it helps to promote the accurate repair, which is very critical for maintaining your DNA strand with the correct building blocks. And that's really important for both for survival, but also for avoidance of cancer. Okay, so these are what I would call the canonical roles. Oh, and I should just go back for the AOA1 family, those connections to cancer probably that's not happening. You don't have that problem with when you don't have AOA1 aprotaxin, because it's much more focused on just the repair. So these are the canonical, what I call the canonical roles of ATM. That's network of responses to double-strand breaks and also sensing your levels of ROS damage. That definitely underlies the dramatic radiation sensitivity we know about. It's pretty much underlying the cancer predisposition and the immunodeficiency, but I'll talk a bit more about those another week. But the question is this the cause of the neurodegeneration? And there's what I'm going to tell you now is there's some lack of linkage there. And that's been a big problem for us in trying to work out what's actually happening in the neurons. So the region in the brain, which is being lost really in AT individuals, is this, the base of the cerebellum, which is sort of at the base of the head here. And within the cerebellum, there's the Purkinje cells, which are especially vulnerable. And this is the region which is progressively lost. So the questions are, is there a specific developmental stage when ATM is required, or is it just stochastic and happening post-birth? Why is it that Purkinje cells in particular, the other regions of the brain don't seem to be particularly affected? Our skin cells, for instance, there's a bit of aging, but they're not affected. Why? What is it? What is special and unique about this region here? And is it the response to double-strand breaks, or is there something else going on? So to talk about neuronal development, we can think of 3 stages. And I should say I'm not a neurobiologist, so this is all thinking about it from the concept of ATM. There's massive neuronal development that occurs prenatally. So when you think about it, kids are born with relatively quite large heads compared to what they're going to be when they're an adult, compared to the limbs, which are going to grow hugely. So there's a lot of prenatal development. So there's one stage prenatally when there's a lot of cell division and replication taking place. There's also a postnatal stage, which is being increasingly recognized, where there's quite a lot of rapid cell division. And that is particularly important for the cerebellum. It's where some of the cerebellum stem cells can start to divide. But the neurons and the Purkinje cells are non-dividing. They've stopped all cell division. They're what we call resting cells, G0. They've gone out of their cell cycle. They're not replicating. So the first slightly odd thing is that other disorders with double-strand break repair deficiencies show microcephaly. I'll explain to you what that is, but it's small head circumference, but they don't show progressive ataxia. Now, I should just say, this process of rejoining your double-strand break is essential. So all the individuals are a bit more like AT variants. They all have some residual activity. But nonetheless, these individuals do not show progressive ataxia. And what I mean by microcephaly is it's a measure of the head circumference, and you can take measure from behind the ears. And the reason why it's small is that you're lacking the forebrain, and it's this region which is particularly small in these individuals. And because of that, these individuals often have pronounced noses, because effectively it's distorted a bit because you haven't got this part of your forehead. And that clearly is not resembling AT in any shape or form. The mouse models for these hypomorphic, the mouse models that kind of resemble these patients, that are models of those patients, show that you've got massive cell death, and it's that cell death pathway that's regulated by ATM, and that it's called apoptosis in the embryos at a stage when you've got this rapid replication that's occurring here. And that microcephaly is present at birth, but it's not progressive. So it's arising postnatally. So as I say, it's very distinct to the situation in AT. So I think that there's a lack of correlation there with this double-strand break repair. Now, the only caveat is that with AT cells, if they're lacking that cell death pathway, then it could be that those damaged cells persist and you don't get rid of them, and then those damaged cells are slowly dying post-birth. But I think it's unlikely to be happening here because it's, remember, the region of the cerebellum is down here, so it's a very different region of the brain. There is another stage, like a bit like this, which is post-birth, neonatal. But again, the other double-strand break repair defects do not show ataxia or Purkinje cell problems. So I think it's hard to really argue that it's a double-strand break repair defect that is exactly correlating with what's happening in the progressive ataxia. And on top of that, there's another strange correlation, which is that the disorders displaying ataxia are much more associated with defects in single-strand break, so just one of those strands of your DNA, than they are with double-strand break repair responses. I've already told you about the microcephaly patients. But these disorders are all involved a bit more in single-strand, well, some of them much more in single-strand break processing than double-strand breaks. AOA1, as I said, does both single-strand and double-strand breaks, but there's many more single-strand breaks than double-strand breaks. So I think probably this is the more important role. But this protein in particular is much more involved in single-strand break repair, really not anything to do with double-strand breaks. And these disorders can all activate that cell death pathway. So although that doesn't mean to say that couldn't be contributing to the ataxia in AT, it certainly says it doesn't have to be the sole reason for ataxia. So generally, I tend to favor a stochastic and ongoing process that happens post-birth that is connected with single-strand breaks or transcription. We'll come back to that in some way, rather than to something that may have happened earlier in development. But I think we absolutely can't rule out that there isn't some impact there. So let's think about the essential features of the cerebellum and the Purkinje cells. What's different about these cells to, for instance, the cells of our skin? I told you they're non-dividing and they're long-lived. And that's quite important because it means if there's damage accumulating, it could have impacts over a long period of time. The Purkinje cells are kind of input signals and then output signals. Again, I'm not a neurobiologist, but they're very metabolically, the consequence of all this is that they're very metabolically active. They have high energy consumption and they have high levels of these reactive oxygen species, mainly because they're doing so many activities in the cell. They're also very transcriptionally active, i.e. they've got a lot of this unwinding here going on, and that's because they're making many proteins. And probably to enable this to happen, they've got a unique feature where they're very open because they want to be doing a lot of transcription. And that, of course, makes them uniquely susceptible to this, what we call ROS damage, to any damage that may be going on here. And ATM, I told you as well, is regulating antioxidants. So, yeah, so I think it's something connected with something more connected with single-strand break repair and this active ability to maintain the active transcription, which and deal with that DNA damage that is particularly, for which these cells are particularly vulnerable, given the whole nature of their metabolic activity and the fact that they're long-lived. So just to then try and summarize what I've told you, there are some additional, so I should say right at the beginning, there are some additional features of Purkinje cells that I will try and discuss next time. I just didn't want to talk for too long and I hadn't had time to go through them, but that kind of all adds to the picture. But what I've told you is ATM is a hub assisting double-strand break repair pathways and it tags key players in the process and it changes their function. And I've told you that we get a lot of DNA damage, both from our own metabolism, from external agents, but also from some of these very complex reactions that are taking place. And Purkinje cells, the link to so this defect in double-strand break repair very much explains the X-ray sensitivity, the immunodeficiency and the cancer. But the link with the Purkinje cells and death is a little bit less clear and it's been a confounding issue for many years. But I think more and more it's being recognized that there's a connection with some of the damage that may arise during that active transcription. And remember, I told you also that if you do have a double-strand break somewhere else, it stops the transcription machinery coming along so that it will crash with the repair machinery. So it's stopping severe accidents happening. And so Purkinje cells may be hyper-dependent on this ATM hub. And that's required to maintain the high transcription levels, which is critical for ATs. So I think some of the questions we want to ask are, how related is the neurodegeneration in AT to some of these other syndromes? Because that could give us, make us have a feel for what absolutely might be defective in AT. We don't understand everything, and you might feel a bit in despair after what I've told you, and you think we don't know kind of how can we treat this, but there are treatments, and I'll go through those next week, the things that we can be doing to nevertheless slow down the demise in AT. Okay, so I'll stop there.